Assalamu alaikum. This is Physiology lecture number 8. In the respiratory module, it is about the transport of oxygen by the blood. The lecture objectives: contrast the tense and relaxed forms of hemoglobin, contrast the oxygen binding curve for myoglobin and hemoglobin, draw an equilibrium that shows the effect of oxygen binding on the relative amount of tense and relaxed forms, describe the forms by which oxygen is transported by the blood, discuss the oxygen hemoglobin dissociation curve, describe factors affecting oxygen hemoglobin dissociation curve, and lastly describe the shift of oxygen hemoglobin dissociation curve during exercise. Before discussing all the detailed points of the objectives, it is important to remind you of the general notes. First, you need to remember that if the diffusion coefficient for oxygen is considered as 1, then relative diffusion coefficient for carbon dioxide is little bit more than 20. Also remember that normally the major limitation to the movement of gases in tissues is the diffusion rate through tissue water instead of through the cell membrane, including the alveolo-capillary membrane, as both oxygen and carbon dioxide are highly lipid soluble. Allah subhanahu wa ta'ala made oxygen of low solubility in water, and therefore it was essential to have a carrier for the oxygen, and that carrier is simply the hemoglobin. Hemoglobin in the RBCs allows the blood to transport 30 to 100 times as much oxygen as could be transported in the form of dissolved oxygen in water. Usually the body needs 230 to 250 ml of oxygen per minute at rest. Also remember that the partial pressure of oxygen in the alveolar capillaries is about 104 or closely to 100 millimeter of mercury. However, the partial pressure in the left ventricular blood is about 95 millimeter mercury due to venous admixture of the blood. Definitely you will ask now, what is the venous admixture? To explain this, I would like you to know that not all the blood reaches the left atrium comes from the alveolar capillaries. Some deoxygenated blood returns to the left atrium from the venous side of the bronchial system. You know the bronchial system receives arterial blood from the bronchial artery, and the veins of this system will return simply to the left atrium. Also, there is some blood comes from the dead space in the non-aerated alveoli, that's to say alveolar dead space. When this venous blood is mixed with the oxygenated blood comes from the alveoli, simply the partial pressure of oxygen will be reduced from the value of 104 to 95 millimeter mercury at the left atrium. Normally, 97% of the oxygen transported from the lung to the tissues is carried combined with the hemoglobin in the RBCs. The remaining 3% is transported in the dissolved state in the water. As you may know from biochemistry, hemoglobin is a protein made up of 4 subunits, each of which contains a heme moiety attached to a polypeptide chain. Each heme has 1 atom of ferrous iron. Therefore, each 1 molecule of hemoglobin has 4 atoms of ferrous iron and can carry therefore 4 molecules of oxygen. In deoxyhemoglobin, the globin units are said to be in a tense configuration. This means there is a reduced affinity to oxygen molecule. When oxygen is first bound, the bonds holding the globin units are released, and this leads to the formation of relaxed configuration. This will expose more oxygen binding sites, and this will lead to more oxygen affinity. This simply means that the more oxygen attachment to the molecule of hemoglobin, the easier will be to attach extra oxygen. Hemoglobin physiologically has 4 functions. The first function is to facilitate oxygen transport, as you may know. The second function is to facilitate carbon dioxide transport as well. The third function, it acts as a buffer to maintain the pH of the blood. I think you should know that hemoglobin is negatively charged, and therefore it can attract hydrogen ion, and this will buffer the extra acidity or the extra hydrogen ion formation in the body. It's one of the most important intracellular buffers in the vascular system. The fourth function of hemoglobin is to transport nitrous oxide that promotes vasodilation. As I mentioned before, 97% of oxygen is transported by the hemoglobin, and only 3% is transported as dissolved oxygen in the plasma. For you as a future doctors, you need to know that giving oxygen to the patient by mask will not increase the carrying capacity of oxygen by the hemoglobin, but will only increase the dissolved oxygen carried by the plasma. To understand this important and crucial point, you need to understand in details the oxygen hemoglobin dissociation curve. Oxygen hemoglobin dissociation curve. This curve demonstrates the relationship between the percentage of hemoglobin saturation with oxygen and partial pressure of oxygen. The curve is sigmoid or S in shape due to the tense relaxed interconversion. The curve shows the following facts. This is the oxygen hemoglobin dissociation curve. It is S in shape, as you can see. The x-axis represents the partial pressure of oxygen, and the y-axis represents saturation. The y-axis on the right side represents the carrying capacity, which is volume percent. It means simply that if the blood is fully saturated with hemoglobin, 100 ml of blood can carry 20 ml of oxygen. And on the right side, the y-axis represents the hemoglobin saturation percent, and the range, as you can see, is from 0 to 100%. Please note that the oxygen saturation is 97% on the arterial side, and it's 75% saturation for the venous blood. And if the blood is fully saturated, as I said, at the partial pressure of 100 millimeter mercury. For the oxygen, the blood will carry 20 mils of oxygen. This oxygen hemoglobin dissociation curve demonstrates the actual values for many parameters in our body. Please note that a partial pressure of oxygen of 97 it corresponds to oxygen saturation of the hemoglobin of 19.4, and at the partial pressure of 40 millimeter mercury for the oxygen, that's to say what is present in the venous blood, the hemoglobin saturation with oxygen is 14.4 mil oxygen for every 100 mil of blood. Therefore, 5 mils of oxygen transported by the lung to the tissue by each 100 mil of blood. And this is going on and on in our body circulation. And please note this important observation from this curve as well. If you look at the range from 60 millimeter mercury for the partial pressure of oxygen up to 100 millimeter of mercury, you can see that the change in the oxygen saturation of the hemoglobin is minimal. This means that if the oxygen partial pressure in the alveoli drops even to a lower value as 60 millimeter mercury, this will not greatly affect the saturation of the hemoglobin with the oxygen. This means the hemoglobin can buffer the effect of hypoxia, and we can stand hypoxia to the level of a pressure of 60 millimeter mercury of oxygen within the alveoli. Factors that shift the oxygen hemoglobin dissociation curve. I would like you to know that some factors shifts the curve to the right and other factors shifts the curve to the left. If the curve shifts to the right, this means that the hemoglobin will hold less oxygen, whereas shifting the curve to the left means the hemoglobin dissociation curve will hold more oxygen for the same partial pressure of oxygen. You need to know that there are three major factors that shift the curve to the right. These are the Bohr effect, the effect of 2,3 dpg, and temperature. I would like to know an important note. If you would like to know if the curve shifts to the right or it shifts to the left, you need to know the effect of P50. Definitely you will ask now, what is the P50? P50 is a convenient index to study the shift. The P50 simply is the level of partial pressure where hemoglobin is half saturated with oxygen. And the higher the P50, the lower will be the affinity of the hemoglobin for holding the oxygen. The normal P50 for arterial blood is 26 to 28 millimeter mercury. Let's start with the Bohr effect. The Bohr effect simply refers to the effect of carbon dioxide and hydrogen ion concentration on the oxygen dissociation curve. Bohr effect equals now the or Bohr effect states that the decrease in oxygen affinity of hemoglobin when the pH of blood falls. Now, an increase in carbon dioxide and an increase in the hydrogen ion concentration, this will shift the curve to the right. By this, more oxygen will be released from the hemoglobin, because by increasing the carbon dioxide and the hydrogen ion concentration, the hemoglobin will hold less oxygen for the same partial pressure. Bohr effect causes left shift of the curve in the lung, because simply in the lung the carbon dioxide will be less and the hydrogen ion concentration will be less, so the curve will be shifted to the left, and this will increase oxygenation of the hemoglobin at the pulmonary site. This figure demonstrates the right shift and the left shift of the oxygen hemoglobin dissociation curve. The normal curve is the one in the middle. I put the arrow. It is the solid line. And the right shift is the line where it is written 7.2 for the pH, and the left shift is the one where the pH is 7.6. Of course, this shift is because of the Bohr effect. As you can see in this diagram, when the acidity increases by increasing the hydrogen ion concentration or by increasing the carbon dioxide, because you should know when carbon dioxide increases, this will make more hydrogen ion within the fluid, simply because the carbon dioxide will unite with water and will make the carbonic acid, and then the carbonic acid will be dissociated into bicarbonate ion and hydrogen ion. Anyhow, once the hydrogen ion concentration increases and the pH will be lower, the curve will be shifted to the right, and vice versa, and that's what happens in the lung, because when we get rid of the carbon dioxide in the lung, the acidity will be less and the curve will shift to the left. Shifting the curve to the right will make it hold less oxygen. And to understand this, please look at number 50 in the x-axis. This is the partial pressure of 50. And if you make a vertical line from number 50 up to the curves, you can see very clearly that when the curve will shift to the right, the saturation will be less, meaning the curve will release more oxygen. And vice versa, when the curve shifts to the left, and if you go by a perpendicular line from number 50 on the x-axis, you will find the saturation will be great. Now, what's the clinical significance of this? What Professor Bohr would like to tell us, that when the blood reaches the tissue, I mean the arterial blood reaches to the periphery and to the tissue, simply there will be liberation of carbon dioxide because of the metabolism from the tissue. The increase of the carbon dioxide at the tissue site simply will shift the curve of the hemoglobin dissociation curve to the right and makes the hemoglobin releases more oxygen, and that's really what the tissue needs. On the contrary, when the blood reaches to the lung, it will get rid of carbon dioxide. The pH will be more, means it will be more alkaline, and this will shift the curve to the left. This will make the hemoglobin able to hold more amount of oxygen when it reaches to the lung. you will apply the same principle for understanding for the shifting to the right of the left. Now, the temperature will shift the curve to the right, and the increase in dpg also will shift the curve to the right. So both the temperature and the dpg will shift the curve to the right. Now we need to explain these other two factors. Now let's speak about the effect of 2,3-DPG. 2,3-DPG is simply 2,3-diphosphoglycerate. DPG is very plentiful in red cells because red blood cells lack mitochondria. It is the product of glycolysis before forming pyruvate. When 2,3-DPG or BPG molecule is then converted to 3PG, ATP is generated. I would like you student to know that some books write the term as 2,3-DPG by considered as diphospho. Other textbook write it as 2,3-BPG and they say it is biphosphate. It's the same compound. Anyway, you should know that when 2,3-DPG or BPG binds to deoxyhemoglobin, it acts to stabilize the low oxygen affinity state or the tense state conformation, making it harder for oxygen to bind hemoglobin and more likely to be released to adjacent tissue. 2,3-BPG acts as such as a part of feedback loop that can help prevent tissue hypoxia in conditions where it is most likely to occur. It's important to note that the behavior of myoglobin doesn't work in the same way as 2,3-DPG has no effect on it. By now, you should know that tissues where 2,3-DPG increases, in these areas the hemoglobin will release more oxygen. Many physiological and clinical states shows that there is an increase in the 2,3-DPG. Let's have some examples. One of the example is in a pregnant woman. In a pregnancy, there is a 30% increase in the intracellular 2,3-BPG. This lowers the maternal hemoglobin affinity for oxygen and therefore allows for more oxygen to be offloaded to the fetus in the maternal uterine arteries. The fetal hemoglobin, that's to say hemoglobin F, has a low sensitivity to the 2,3-BPG. So hemoglobin F has a higher affinity for oxygen. In other words, hemoglobin A, which is the hemoglobin of the mother, will afford more oxygen, while hemoglobin F, which is the hemoglobin of the baby, has higher affinity for the oxygen. Therefore, although the partial pressure of oxygen in the uterine arteries is low, the fetal umbilical arteries, which are deoxygenated, can still get more oxygen from the maternal blood. This slide gives more information about more physiological and clinical cases where the role of 2,3-DPGs has an importance. In thyroid, for the thyroid hormones, the growth hormones and androgens, all these hormones increase the concentration of DPG and the P50 and make the hemoglobin afford more oxygen. Ascent to high altitude, that's to say if we want to go up into a mountain, this will trigger a substantial rise in DPG concentration in the RBCs. DPG also increases in anemia, and as we know, in anemia the body needs oxygen so much because the amount of hemoglobin in the body and the number of RBCs usually are lowered. And in diseases associated with chronic hypoxia, such as airway obstruction or congestive heart failure, in all these cases, DPG keeps the oxygen dissociation curves shifted slightly to the right all the time. In hypoxia, the increased level of DPG increases the oxygen release. This mechanism adapts for hypoxia caused by poor tissue blood flow and hypoxia due to living in high altitude. And this explains how can people live in the mountain and will not suffer from hypoxia simply because their blood and their RBCs have higher amount of 2,3 DPG. The other effect that can shift the oxygen hemoglobin dissociation curve to the right is the temperature, and this is one of the effects usually we see during exercise. So if you would like to understand the physiology of exercise and the behavior of the hemoglobin during exercise, you should know that exercise makes many factors act together to shift the curve to the right. These are, first, an increase in carbon dioxide release by the tissue. Second, the acid formation and the increase the hydrogen concentration in the exercising tissue. Third, there is also an increase in the DPG production during exercise. And last, there is an increase in muscular temperature by 2 to 3 degrees centigrade, and all these factors act together during exercise to shift the oxygen dissociation curve to the right and make the hemoglobin deliver more oxygen at the exercising muscle. For the proper understanding of how oxygen is transported from the blood to the exercising muscle, we need to know few notes about the myoglobin. The myoglobin is a protein found in skeletal muscle. It's also a red protein because it holds iron. It resembles hemoglobin but binds with 1 rather than 4 molecules of oxygen for every molecule of hemoglobin, or 4 moles of oxygen for every mole of hemoglobin. Its dissociation curve is a rectangular hyperbolic rather than a sigmoid curve, and placed to the left of both fetal and adult oxygen hemoglobin dissociation curves when plotted together. As myoglobin has a higher affinity for oxygen, it extracts oxygen from the hemoglobin and delivers it later to the skeletal muscle when oxygen is cut off during skeletal muscle contraction. In other words, it acts as a holder for oxygen inside the skeletal muscle, so it's a reservoir for the oxygen, and once the oxygen is cut off from the hemoglobin, it will deliver the oxygen to the skeletal muscle just to overcome the cut off of oxygen. This diagram shows you the position of the myoglobin, and you can see from the arrow I put that the oxygen dissociation curve for the myoglobin is to the left of both hemoglobin A and hemoglobin F dissociation curves of the oxygen. So from this diagram you can see that hemoglobin A, which is from the mother, usually delivers the oxygen to hemoglobin F, and both of them deliver the oxygen to the myoglobin. In this diagram you can understand why one is to the left of the other. Please look at the arrows in below. The P50 for hemoglobin A is about 27, and to the left of it comes hemoglobin F, and the P50 is 19, and the lowest P50 is for the myoglobin. It is just 2 millimeter mercury pressure. That's why we say that the myoglobin dissociation curve is the left of both curves, and it can extract oxygen from both types of hemoglobin. I hope you understand this lecture. And as usual, this is the test question I give you by the end of this lecture. Try to solve it. If you have any difficulty with understanding or solving this question, please, you can contact me and you can write me by the email. And good luck.